Hi, and welcome to today's lecture of the course Medical Aspects in Microbiology. Today, I'd like you to hear about and learn a strategy by which humans defend themselves against invasion of pathogens, as it occurs, for instance, during bacterial infections. We all know this. We have experienced this in a bacterial infection. Our throat hurts, it is red, the tonsils are swollen and red, and the tongue can be gray and furry. Overall, we were shitty. But what happens actually during that time, during these infections, and how does the host, like we as humans, defend ourselves against invasion of pathogens? One compound I want to introduce is hypochlorous acid, or HOCl. HOCl is the active ingredient of household bleach and pretty well known. Bleach is extensively used in hospital settings as disinfectant in order to prevent bacterial colonization on biotic and abiotic surfaces. Some of you might know this guy here on the left side of the slide. His name is Bleach, and as a character of a Japanese manga series, he's able to defend humans from evil spirits. And I want to focus today a lot on bleach and highlight that even we as humans have bleach factories in our body, which we use actually to defend ourselves against bacteria. And with this, I would like to ask a question. In which cell type do you expect that hypochlorous acid is produced during host defense? Take a minute to think about that, and these are the four options. Number 1, muscle cells. Number 2, in neutrophils. Number 3, erythrocytes. Or 4, thrombocytes. Thank you very much for answering the question. And as most of you guessed right, neutrophils are the cells in which HOCl is produced. But how is this actually going to happen? And this is shown here in the next slide. It begins with the activation of NADPase oxidases, which are shown here in red, that are located in the phagosome membrane and catalyze the reduction of molecular oxygen to superoxide. In the next step, superoxide dismutases, shown here in green, subsequently convert superoxide into H2O2 peroxide, which then functions as a substrate of myeloperoxidases, MPO, shown in blue. Myeloperoxidases are heme-containing haloperoxidases and capable of generating HOCl by catalyzing the reaction of peroxide H2O2 with physiological concentrations of chloride. Once released into the phagosome, HOCl evolves a very rapid toxic effect on bacteria, contributing to the effective killing of invading microbes. But how is actually HOCl affecting? And here we see the amino acid chain, which should illustrate the protein. And protein are known as the workhorses of cells, also in bacteria. And HOCl affects the amino acids of proteins. We differentiate here between reversible and irreversible modifications. the disulfide bond formation, methionine sulfoxide formation, and N-chlorination are reversible protein modifications and often used to regulate protein function in response to oxidative stress. HUCl also causes the oxidation of histidines and tryptophans and the formation of deutyrosines and sulfenic and sulfonic acids, as well as methionine sulfoxamine intermediates, which all are irreversible modifications and lead to protein unfolding, aggregation, and degradation, which in turn leads to the death of the bacteria. So, as a take-home message, I would like to ask you the following questions. How do humans control the bacterial invasion? Thank you very much for the answers. What I want you to take home is that neutrophils produce the highly effective oxidant hypochlorous acid HCl during mammalian host defense, and that HCl is a potent oxidant that modifies macromolecules like proteins and thus causing the death of the pathogens. The last slide shows you where to find the respective literature, and also if still questions are there, I offer an office hour either by appointment or directly after class. Thank you very much.